So moving on to 8.3. So 8.3 deals with some major topics such as sex-linked traits, and also we'll mention, this was actually earlier, but we'll mention some pedigrees, some of those kind of genetic trees in this part as well. So sex-linked traits are a little bit different than what we've seen before, although we can still do Punnett square problems and we can still visualize chromosomes for them. But when you talk about a sex-linked trait, you're talking about a trait that is on one of the sex chromosomes. How many sex chromosomes do you have? 2. You have two sex chromosomes in all of your autosomes. Remember that word, autosomes? How many autosomes do you have? How many total chromosomes do you have? 46, right? You just told me that 2 of those are sex chromosomes, which means that leaves 44 chromosomes, and those are your autosomes. So you have 44 autosomes, or you have 22 pairs of autosomes, or if you're looking at one of those karyotypes, right? The first, and we'll see a karyotype in a minute, the first 22 pairs, the first 44 chromosomes are your autosomes. Chromosome 1 from mom and 1 from dad, 2 from mom, 2 from dad, all the way up to 22 from mom, 22 from dad. And then that last pair, the only pair that can be different from each other, are the sex chromosomes. No, because you have 2 sex chromosomes. You have 23 pairs of chromosomes, so that's 46. I don't know. Okay, so autosomes are your non-sex chromosomes. All right, those should be homologous. So chromosome 1 from dad and chromosome 1 from mom are homologous. Chromosome 21 from mom and 21 from dad are homologous. Now, with sex chromosomes, a little bit different. These were first discovered by Thomas Morgan at Columbia University. You read a little bit about his fruit flies for yesterday, right? He was doing this research with fruit flies, and ultimately what happened is he started to get these, results that weren't really consistent with some of the Mendelian genetics. And so he had to try to figure out kind of a mechanism of why that was happening. And so he was dealing with fruit flies, or Drosophila. Pretty simple experiment, things to work with. In fact, I've been playing around with whether or not we'll do this next semester or not. But fruit flies are pretty easy to work with. They have good genetics. You can have a lot of different traits with them. They reproduce very, very well. They lay little eggs, and then you can kind of clear out the parents before the eggs hatch, and then whatever shows up in your little vial is the offspring. It's pretty easy to feed them. They have 2-week generations, which is pretty good, right? Even better even than our fast plants, it seems like. They have 4 pairs of chromosomes, so they have genetics, but not a huge number to deal with. And they do have male and female. And you can actually tell the difference between a fruit fly from male and female. If you've never done it before, if we don't do it this year, if you take any high school science, they'll probably have you do a lab with it. But you can, there's actually a substance called FlyNap, and it's basically a little chemical that you put in their container, and it knocks them all out. Right? It doesn't kill them, just knocks them all out. You can do a similar thing with cold. So then you can kind of line them up, take them out of the tube and line them up on your desk and put them under a dissecting scope or a stereoscope, kind of like this one right there. And there's some general traits that you can look at for males versus females. The body size is a little bit different. There's some different patterns and stuff on it. And so you can look at the male versus female, which if it's a sex-linked trait, you're going to have to know if it's a male or female. And then you can also look at the eye color. Is it red or is it white? Are the wings fully formed? Or are they kind of ripped, shriveled, and wrinkled? There's some really weird traits out there. There's one where it's kind of like, it's got like almost looks like a leg coming out of the top of its head. And so again, you can do some pretty simple genetics with these guys. So that's what Thomas Morgan was doing as he worked through some of the stuff. Now this picture is a karyotype, right? And so first of all, we can see our 44 autosomes right here, right? 44 autosomes. So 2, 4, 6, 8, 10, 12, all the way up to 44 right there. And then we have our sex chromosomes. This is not covering this one, by the way. The Y is smaller than the X, so male or female? Male. Right, this one's going to be a male because we have XY right there. So all of those guys are autosomal. These two are the sex chromosomes, giving us our 46. In males, this is the only pair of chromosomes that do not have to be homologous. Right? Why aren't these homologous to each other? Not the same size, but we can do better than that.
They don't have the same genes on them.
Good. They don't have the same genes in the same places, right? If I look right down there, and I look right at the bottom of the X, it's going to be a totally different gene. So they are not homologous. So with a female, however, this would be an X and an X, and those X's would be homologous to each other. So really, females have 23 homologous pairs, males have 22 homologous pairs. But we all have 44 autosomes and 2 sex chromosomes. So this is why things start to get a little bit different with males and females, because if you're a male and you are talking about a gene on the X chromosome, you only have one of them. So you can't be homozygous dominant or homozygous recessive or heterozygous. You either have the dominant allele or you have the recessive allele. And whichever one you have is going to show up. But of course, with a female, you have 2 X's, which means you should have 2 genes, which means you should have 2 alleles for those traits. So basically, for females, there's no difference in the whole Punnett square Mendelian genetics, even if it's sex-linked. For males, it is. We'll see some of those as we go through this. So the discovery of sex-linked. So if we kind of look at, so we'll try to designate between our males and our females. And so our females will just do with a pink body. Males will do with a blue body. You can see we're following eye color. There's our nice red eyes. There's our nice white eyes on the male. True breeding, right? So that one only produces red, that one only produces white. Now, if those guys cross, well, based on a simple Punnett square, all right, you would expect 100% of the offspring to be red eyes, regardless of male or female. Because you remember this one would be like big R, right? And this one would be like little r. And so all your offspring would be big R, little r. Well, if it's big R, little r, they would all be red. And then if you took that F1 offspring and you let them cross, now you have big R, little r times big R, little r. Now we'd expect that 1 to 2 to 1 ratio, right? Homozygous dominant, 2 heterozygous for every 1 homozygous recessive. And so we would expect basically the following. So how do we determine this? Okay? How do we determine, or how do we explain that if we're just doing normal kind of Mendelian genetics with dominant recessive, how do we explain that all the females are red, but the males are 50-50? Right? So we're getting some different results here that we got to try to figure out why. So when Morgan took these flies, again, when he started this, he was kind of going off of Mendel's stuff, right? So if we took those 2 flies and fill in their genotypes here, so I was kind of talking through earlier, we would expect all of these middle guys to be heterozygous, right? Big R, little r. And if they were all heterozygous, we'd expect them to see all red eyes, which we see in that idea. Then if we took the F1 generation and crossed them, and taking 2 big R, little r's, crossed them, fill in our Punnett square, we'd expect 25% to be big R, which would be red, 50% to be big R, little r, which would be red, and 25% to be little r, which would be white. Well, there's our 3 red to 1 white, right? Our 3 to 1 phenotypic ratio. The problem is, that's not what Mendel saw, right? He did not get these results. So then the question you got to ask yourself is, well, first of all, what would we do how would we determine that these aren't good results? Well, that's what that chi-square thing we've been doing is, right? You'd look at what numbers did you get, what did you expect, do a statistical test, and basically this would have shown you that your p-value was less than 0.05, right? Less than a 5% chance that it's random, that the differences are by random chance alone, and so you'd have to come up with a different mechanism, a different hypothesis. And so that's what Morgan did. Said, okay, this isn't fitting, there's got to be something else going on here. So how can we explain these results? If there's obviously something different than what was happening in Mendel's pea plants. Now, in humans and other mammals, there are 2 sex chromosomes, the X and the Y. Now, some organisms have a little different, and there's actually some reversals of this and stuff like that. But in general, when we're talking about the 2 X chromosomes, we're talking about the female being XX, having that homologous pair. You have some redundancy. So it's basically acts like those other 22 pairs of autosomes, right? So their 23rd pair of homologous chromosomes there. With a male, you have an X and a Y. So again, we can see the size difference. We'll see this in more detail in a couple minutes, but the Y chromosome not only has different genes, but very few genes on it as well. We'll see the significance of that. It's going to develop as a male if you're an X and Y. You have no redundancy. You only have 1 allele for each of those traits. And so, when we do a Punnett square, right, males go on the top of our Punnett square. We got an XY, female on the left, XX. And so this kind of explains our probability of getting a male or a female. which I think most of you know it's 50-50, but this kind of shows you how that works, and it's going to be important for our probability stuff tomorrow. So a male and a female should have a 50% chance of having a girl and a 50% chance of having a boy. Right? So 50-50, just based on your sex chromosomes alone. But then we got to remember, okay, so if we actually want to follow a trait that's on a sex chromosome, and typically we're going to follow a trait that's on the X chromosome. Oftentimes we'll call it an X-linked trait, because that specifically tells us it's on the X chromosome, as opposed to sex-linked kind of leaves it vague. Is it on the X chromosome, the Y chromosome, etc. So all we have to do is take this Punnett square right here and say, oh, so we're following the gene for hemophilia. That's on the X chromosome. And so we'll have to still write our X and Y's because we have to track which chromosomes in males and females, but we'll also add the allele, the letter, right, or the genotype to our X and Y's as well, and we'll just fill out our Punnett square the same. So if we relook at Morgan's flies, right, we had this cross here. Well, what if we now track the X and the Y's along with the actual alleles of the genes? And so we said earlier that the red-eyed fly was big R, right, because it was true breeding. But now we're just following the fact that, okay, this is X-linked, or if this is on the X chromosome, this X chromosome has the dominant R, this X chromosome has the dominant R. Now the male, we said, was white eyes, right? So the male has to have R. Now earlier we had said it was going to be little r, little r. Why can't it be little r, little r? Good, only one X chromosome, right? And so the X chromosome, sure, it has a little R on it, but then it has a Y. And since these are not homologous, the Y does not have that same gene, so you can't put an R on that gene, right? So now you see, okay, so it only got one copy. It got one copy of the recessive allele, but having only one copy and no other copy, right, this guy becomes recessive. So now we just fill in our Punnett square. The male on the top here, X little r, Y. Female on the left, X big R. And we just fill in our Punnett square. Again, we're always doing the capital letters first, keeping the X and Y's. So two things that this shows. One, it shows your 50-50 split of male and female, right? So here's my 50% chance of females. Here's my 50% chance of males. But it also tells you in terms of this trait, red eyes versus white eyes on a fruit fly, what is this one going to be? Red-eyed female, right? Heterozygous red-eyed female. So that's why pink body, right? Red eyes. This one's going to be red-eyed male. And again, there's no homozygous or heterozygous. It's just dominant. This one's going to be red-eyed female. This one's going to be red-eyed male. So that looked somewhat similar maybe to what we had before. But now let's take two of these. We only have one type of female and one type of male to choose. So let's take two of those and cross them. So we're going to take a heterozygous female with a recessive male, with a dominant male. Set up another Punnett square. Male on the top, X big R, Y. X big R, X little r. With the female on the left. And we fill them in. X big R. Red-eyed, homozygous, dominant female. Big R, big Y. Red-eyed male. Big R, X little r. Red-eyed female, just heterozygous, but it's going to look phenotypically identical, right? And X little r, Y. White-eyed male, right? So how many different phenotypes? Three, right? You have a red-eyed female and a red-eyed male. Those are different phenotypes, right? If you're a male versus a female, it's a phenotype. And you have a white-eyed male. So three phenotypes, four different genotypes from that cross, right? And so we have 100% red for females. And typically, we need to kind of record males and females differently. So don't say 3/4 red, right? You're going to say, okay, so what do I have going on with the females? Oh, 100% red, 2 out of 2. What do I have going on with the males? Half red, half white, right? So there's our three different phenotypes split up. Now, these are the results that were similar, or that were statistically similar to what Morgan was finding, right? So it explained this pattern of inheritance, or this idea of a sex chromosome different between males and females, was supported by his data. So a little bit more about this X and Y chromosome. So the Y chromosome has only a few genes on it. It has a gene called the SRY gene, which is the sex-determining region on the Y chromosome. The sex-determining region on the Y chromosome, SRY gene. Kind of an important gene. Why wouldn't you expect the Y chromosome to have a whole lot on it?
Why wouldn't you?
Why wouldn't you?
Well, because only the guy has it.
Okay, only the guy has it, right? So it'd be kind of weird if like something really important was on the Y chromosome, right? Like the gene for eyeballs or something like that. And eyeballs are more important or more complicated than a single gene. But right, if it was only on the Y chromosome, that would mean only the guy would have it, and all the females out there wouldn't, right? So basically, the sex-determining region on the Y chromosome is basically only going to have genes for maleness, okay? Genes for maleness, for male development. And if you lack the Y chromosome, you lack the SRY gene, which means you're not going to develop male traits, okay? And so basically, what's going on there is if you have the gene, you become male. If you don't have the gene, kind of by default, you become female. All right? So if you have the gene, you become male. If you don't have the gene, you don't make the proteins. If you don't make the proteins, you go and you become a female instead. Okay? So it's basically like the regulator for maleness, turns on genes for the production of male hormones. Male hormones, of course, then cause that whole, right, hormones we talked about, signal transduction pathways. That's going to cause the whole cascade of other signals and effects to make other proteins and things like that. And that's going to be happening during that developmental stage, right, as the fetus is developing inside its mom. And again, many different effects from that gene, so pleiotropy. Yes.
You know how they like say that like being gay can be due to like your predisposition or whatever of genetics? Does that deal with this at all? Like those hormones not being fully developed or something? Maybe?
That I don't know. I don't know how much they would even know on that. So I would think if it has to do something with genetics, it seems like it would be a good guess that it has something to do with that. But it do not know that. Yes.
What about like the people who have more Hemophilia traits?
There are some problems. If you think about, remember non-disjunction? When chromosomes fail to separate correctly, which could happen during a meiosis 1, where if the tetrad did not separate into the two homologous pairs, one of the tetrads might have gotten pulled to one side, and none of that chromosome got to the other side. And make sure you understand non-disjunction. You kind of go through it and say, okay, what if non-disjunction happened for this chromosome in meiosis 1? What would my 4 final cells look like in terms of missing and gaining a chromosome? And it can also happen instead in meiosis 2, where you have the homologous pair lined up, right? And it's supposed to separate into the sister chromatids, right? If that homologous pair doesn't get separated correctly, that whole pair goes to one side and 0 goes to the other. And so ultimately what happens, if the problem happens in meiosis 1, before the cell divides even once, all 4 of your sex cells will have the wrong number. 2 of them will be 1 too many and 2 of them will be 1 too few. If it happens in meiosis 2, anaphase 2, that separates, then 2 of your cells, 2 of the sex cells being produced would be normal, and 1 of them would have 1 too many and 1 of them would have 1 too few. So make sure you go back to that non-disjunction, you understand that idea. So the reason why this is for that question is that the sex chromosomes have to get separated during meiosis as well, right? They form a tetrad and they get separated. And so there are conditions where there's a condition XXX, where a female gets 3 X chromosomes. There's XYY, where a male gets an extra Y chromosome. It used to be believed that led that a higher proportion of inmates were XYY, and the theory was more Y chromosome, more testosterone, more stupid acting maleness. I don't know, but that turns out, I believe, to have not been true. There's XXY, right? And that's where you get into, it is a male because it has a Y chromosome and the sex-determining region on the Y, but having the XX, you kind of get the double dose of some of the female hormones, and that's where oftentimes you'll get a male, but they'll have some of the female secondary sex characteristics. Not hermaphroditic. I don't know quite how that's caused.
Wait, so what does XYY do?
XYY
would probably lead to extra testosterone. I believe they'll
But they wouldn't be dysfunctional or anything. You wouldn't be able to tell.
I'm not sure too much on that, but I believe both XYY and XXY and XXX. I think all 3 of those, and you can actually have XO. just one sex chromosome, just an X, and the O means that there's nothing else there, so 45 chromosomes.
So you're neither a boy or a girl?
So that would be a girl, because you don't have a Y chromosome. If you don't have a Y chromosome, by default, you're a girl. That scenario, I think most of those scenarios where your sex chromosome numbers are not the normal 2 of them, I believe you cannot reproduce. I don't think you can produce offspring. Yeah.
What about in that case of that runner that was in the Olympics? I can't remember her name.
Yeah, I know who you're talking about.
I didn't know if she should race with boys or girls.
Yeah, I can't remember what the basis of that was, though.
They didn't believe her that she was a girl, so they made her undergo gender.
Yeah, and I don't know. I mean, she
had the thing where she had both levels of testosterone.
So I'm assuming they tested the chromosomes. I don't know enough about it.
She had 3 times the level of testosterone.
Right, but I don't know if that meant that she had a Y chromosome, because I would think if she had a Y chromosome, then they wouldn't have considered her female.
I do think that they.
But there's other reasons.
Her a hermaphrodite.
Yeah. So I don't know. I never saw the conclusion of that, so we'd have to look that up.
So then, shouldn't she just get to pick then what she wants to
She ran with
maybe.
girls.
She just said something like, "Oh, it's like how God made me," and that's okay.
So what's on the X chromosome?
The X chromosome has just normal everyday genes. Now there should be. I would assume that there's some other. I don't really want to say female characteristics, but for the most part, it's just your everyday genes. For example, there's a gene that allows your blood to clot. And if you don't have that gene, if you have the recessive gene, that's what hemophilia is. And so a male only has one copy of that, and if they have the recessive one, they're hemophiliac. If females, it's much more rare because they have to have 2 copies of it. And so there's just a lot of normal everyday genes, just like the other 23.
How does that female?
The femaleness, so dealing with the estrogen production would probably be.
That develops later in life, though.
Yeah, I would assume that has something to do with that chromosome, and males would have it too, but probably the signals would be producing the testosterone instead. I'm not an expert on that. Yeah, last question. We got to keep going.
What about Jamie Lee Curtis? Like, doesn't she? What? No, I'm serious. She has this issue. She has testosterone and estrogen. What would that be?
I don't know enough about. I know her, but I don't know. We'd have to look that up too and see how it applies.
What does SRY mean?
What's that? Sex determining region on the Y chromosome. SRY, sex determining region, Y chromosome. All right. So the X chromosome, and maybe Chloe, this answers your question. Other genetic or traits beyond sex determination. Mutations. Remember, mutations are kind of have the non-normal type. Can lead to things like hemophilia, muscular dystrophy, color blindness. We'll talk about 2 of the 3 of those coming up. But males are much more. It's much more common for a male to be colorblind because, again, they only have 1 X chromosome, and so if they get this recessive or this mutated chromosome on their X, well, now they're colorblind. For a female, they would have to inherit 2 X chromosomes with a mutated copy on it, or have 2 mutations happen, and the odds are much, much less. Same thing with hemophilia, and muscular dystrophy makes a protein, or the gene is supposed to make a protein. With muscular dystrophy, that protein is not being produced, and basically your muscles aren't able to function how they're supposed to be. Hemophilia, the normal gene makes a factor that causes your blood to clot. So you get a little cut, that blood clots to plug up the hole, right? Well, if you have the mutated gene, your blood doesn't clot like it's supposed to, and you keep bleeding and bleeding and bleeding. And you'd be like, well, wouldn't you go to the emergency room? And yes, probably you would, unless you don't know that you're bleeding internally.
Does
Right?
Weir's have that?
What's that?
Yeah, he does, so it can't be in the
Could be. Yeah. So again, there are females with each of those, but much, much less common. So here's X chromosome. You can see this is just a sampling of what's on the X chromosome. There's a lot of different things. So a lot of just kind of, well, you'd have to really read it through carefully to see. But if one of these genes would be considered to be X-linked because it's on the X chromosome.
It looks like most of those are just like syndromes.
Yeah, well, that's what this one's focusing on. Because this idea right here, more than 60 diseases are traced to the genes on the X chromosome. But keep in mind, there's tons of things that can go wrong with your other chromosomes, and there's a lot of other conditions caused by your non-sex chromosome. So, yeah, they're mostly showing you, I mean, there's going to be about 1,000 genes on this X chromosome. This is definitely not showing 1,000 things, right? It's definitely focusing on the things that can go bad and not kind of the everyday things.
Do you have a map of all of what's
Yeah, I can show you that tomorrow if you ask me at the beginning. Here's that Y chromosome, much, much smaller. Only about 30 genes or less on the Y chromosome. Remember, your average gene, your average chromosome, I should say, has about 1,000 genes. Y chromosome, less than 30. It's got the sex-determining region, which is the biggest thing that we're going for. I don't know what half these things mean, and I don't know how...
Air guitar.
I don't think they're wait, why would there be a sex-determining region if when you have the Y chromosome?
What is the air? What is the air?
Because that it has to do something. It has to have directions somewhere. This says the directions.
Channel flipping, casting
Okay, please don't all talk at once. We got 1 minute. Stop talking, please.
But like, if they didn't have that sex-determining region, would that not be considered a Y chromosome?
Well, if they were missing that, they wouldn't have the stuff to make a male. I mean, just having the chromosome, if you just say, well, you have the Y chromosome, you're going to become a male, your chromosomes have genes on them. You can't just, if you have a chromosome that doesn't have the genes, you don't have
Well,
a
I know,
chromosome.
but then that's not just a specific region, is it the whole chromosome?
No, it's a specific region.
Itchy.
It's a gene on the Y chromosome. Your Y chromosome does more than just that. That's the major importance of the Y chromosome. You have to have this gene, and this gene determines maleness and creates the hormones to create a male. If you don't have that gene, you don't have a male, but there's a few other things on there as well. Okay? All right, so first of all, this idea of pedigrees. So we see Queen Victoria. Some of you might know this story, but this is looking at hemophilia. And we mentioned yesterday that hemophilia is caused by a sex-linked trait. More specifically, it's an X-linked trait because it's on the X chromosome. And what happens is normally you have a gene that codes for a clotting factor. The protein that's produced by that gene helps your blood to clot when an injury happens. And so, if you're a female, you have 2 copies of that. So if one of your copies happens to not be the dominant form or the wild-type form, in other words, it's not making the functional protein, you still have the other gene that's going to make the functional protein. Well, for guys, you only get 1 copy of it because you only have 1 X chromosome, and so if you lose it, or if you don't have the right copy, or if your copy doesn't make the protein, you're missing that clotting factor, and you continue to bleed. So anyways, there's been a lot of stats done on Queen Victoria and on her descendants and kind of this progression of hemophilia through their family. And I thought I kind of had it up there so that picture went away, but I guess I don't. And so, first of all, kind of in the background here, pedigree was mentioned earlier in the chapter, and we never really talked about it. But someone remind me what a circle is in a pedigree?
Man.
Woman. Right? Female. So you remember this? You read about it. Squares are men. If it's connected horizontally, they're married. If it's connected, well, generation would be vertical, so this would be generation 1 and 2 and 3 and 4 and 5 and 6 and 7 and 8 and 9 generations listed there. If you have kind of a horizontally connected above them, these would all be what?
Is there inbreeding in their family?
Siblings. What's that?
Is there inbreeding in their family?
Well, it would sure look like Prince Albert and Queen Victoria were a little bit scandalous, perhaps. Yeah.
What's the
They had a lot of kids.
What's that?
Wait, they were
bend
cousins?
above the Duke of...
The bend right there.
Was that like cousin?
That.
Prince Albert came from just a man.
I'm not quite sure.
Is that 2 family lineages? Awesome.
Yeah, I honestly am not quite sure there because you got
I know, why is there just a man who had
with
kids?
this. So I don't know what that one is. I think it's just crossing over. I think you're right that they're putting here's a family lineage and here's a family lineage, and they're trying to get this guy in here to marry poor Alice, but it's actually coming from this. So I think that's showing that hey, don't confuse this with one of their kids. It's actually from this family over there. But the point is, you can look through this and you can start to figure out what was happening. And in this case, the shaded ones is typically what we're following. So the shaded people would have the hemophilia. What does it mean if you have a little half-shaded carrier? Why don't you see any half-shaded males?
Because if you have it, you have it.
Because in sex-linked traits, a male can't be a carrier, right? They only have 1 copy, so you either have it or you don't. And so carriers typically applies to a sex-linked trait or oftentimes they're called a heterozygous carrier, I think is what your book listed them as, right? And so we can use this idea of a pedigree to kind of track a different trait or characteristic throughout. So there we go. So again, you can see that there's a lot of hemophilia kind of getting trapped inside of there, right? As you kind of go through that family. Notice a lot of males, right? Male, 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 right? Lots of males with hemophilia because, again, there's such a greater chance of them having just one bad copy, and the odds of a female getting two bad copies is just a lot slimmer. And so you look at that and you say, okay, well, how do I know it's actually a recessive trait? Well, if it was a dominant trait, let's see, how can we figure that out? If it was a dominant trait,
then more
that would
women
mean
have
that
it, right?
these two people, first of all, if it was a dominant trait, you wouldn't have any half circles, right? Because you would have it. So pretend this is white, this is white. Because sometimes they don't show carriers. If these two people were white and hemophilia was a dominant trait, both of these people would be what? Homozygous, recessive, which would mean all little letters, right? If they have all little letters, could they produce a child with a dominant? No. And so that's some different patterns we can look at. And actually your book did a nice job, and you might want to go back and read it if you just don't feel like you're that familiar with pedigrees, because there is a pedigree on the test tomorrow. And your book gave 2 steps, and I actually didn't ask the question very well on your review sheet or on your reading guide, but it said, okay, if it's a dominant trait, what are 2 things to look for? And then it said, okay, what if you're following a recessive trait? What are 2 things, 2 keys to look for in your pedigree? And I think that was in the end of 8.1.
Is the one on the test this big, or is it?
No, it's not this big.
Okay.
Not this big generations. We'll come back and I'll show you 2 easier Punnett squares in a minute.
How do they know those women carried it?
What's that?
How do they know those women carried it?
That's a good question. I mean, some of them they don't really know, and so they did the question marks. Other ones, I think it's based on the fact that we know he doesn't have it, yet they had a son that did, so how did that son get it?
Yeah.
Got it from his mom, right? And again, that's another thing to point out. All males get their X chromosome from mom, because their dad gave them the Y chromosome, right? And so again, it would be the mom that would pass it down to the sons. So again, you can kind of backtrack and say, well, she must have been a carrier because she didn't have it, but she produced a son that did. So again, our hemophilia, we can just do the same thing, right? Here's a female. What would we call her? Heterozygous or carrier. That big H, little h. There's a male. Does he have it?
Yes.
No.
No, it's a recessive trait, sex-linked recessive, so he's just normal. So the female would produce, half her eggs would have the capital H be normal, half her eggs would have the lowercase h and produce hemophilia. For this male, half his sperm is going to have normal hemophilia gene, and half his sperm is going to have the Y, right? And so again, that was segregation right there, right? You take your genotype and they get segregated into different gametes. So 50%, 50%, 50%. We could plug that into our Punnett square if we wanted, which is really what your Punnett square is doing, the same thing. And that those people, a carrier female and a normal male, have this ratio. They have a 50% chance, and it's kind of up to you on how you want to decode this. You could say they have a 100% chance of having a normal female. That might be misleading though, because although they both, neither of them have hemophilia, it's really a 50% chance of females that are normal and a 50% chance of females that are carrier. There's a 50% chance of males normal and a 50% chance of males with hemophilia. And typically with sex-linked, you want to divide up the genders. Say X number of females will have this, X number of males will have that.
If a recessive male has a kid with a heterozygous female, then you could produce a female child with hemophilia?
Yes.
Okay.
So to have a female with a sex-linked trait, first of all, the father would have to have it, and the mother would at least have to be a carrier.
Okay.
And then I think there's like a 50-50 chance. So, anyways, the carrier had the disease. This is kind of an interesting thing, and I only want to mention it in passing. This will not appear on your test tomorrow, okay? But it's something that a lot of people don't know about. That females have 2 chromosomes. Now that's not shocking news, okay? Females have 2 X chromosomes. However, 1 X chromosome becomes inactivated. Now you might say, well, how is that different than a male who only has 1? And the difference is it's random which chromosome gets inactivated early on in development. And so you can kind of see what's going on here. So this one, right, you have two X's, the orange and the black one. In some cells, the black one stayed activated, and in some cells, the orange one stayed activated. And then at that point, every time they reproduce and reproduce and reproduce, if it had the orange X and the other one kind of shriveled up, the orange X would get copied, and not the other one just wouldn't be functional. And so as you kind of expand this into an organism, you can see that some of them are going to have the one X, and some of them are going to have the other X. And so it's kind of a weird idea. It actually, a calico cat, if you ever thought about why it has these spots, is because of this X inactivation. And so depending on where in the body the certain X is inactivated, it leads to the different colors. The other one, this little circle here, is called a bar body. So it's basically a condensed X chromosome that just kind of is there, but it's not readable. You can't use it to read the genes. So any information into those genes in that cell is only going to come off of that X and not that X. But it's not the same as males because they do have the other X, and some of their cells, that other X is activated. And so you think about like hemophilia. In other words, what that would mean is that some cells in a carrier female, some cells would not be producing the correct protein, but other cells would be. Right? Does that make sense? So some cells are producing the right one, some wouldn't be if it's a carrier, but overall, since some cells are doing the right thing, you typically don't see many effects from it. And so it's a random event. It causes this mosaic in your book. Actually, I don't know if your book has it. I think it's the other book. Kind of shows it. It's called a mosaic because if you actually like look at a cell that would do this for a female, you might have a patch of cells up here with one X activated, and the patch over here, the other X is activated, and over here it's the other one again, and here it's the first one. And so it's kind of this patchwork where in a calico cat that leads to the different colors.
Is that why you don't really like have male calico cats?
Yes. Very good.
Then why is it that not all female cats?
That is a good question. I don't know that answer.
Different breeds.
So anyway, it's kind of an interesting thing, but again, we're not going to talk about X inactivation, and so we'll kind of move on. Male pattern baldness, which I won't go into too much detail. All right, I was going to put my head on his body, but I decided not to. It's a little bit different. It's more of a sex influence trait, so it's not. It still is just dominant recessive stuff, but again, a little bit different. Being it's an autosomal trait, which means it's on one of those first 44 chromosomes, the first 22 pairs, right? But it's affected by some of the hormones that are created on the sex chromosome. So kind of an interesting thing there. Again, we're kind of just out of time, and that's not going to be on your test tomorrow either. Now finally, a little pedigree analysis. Probably should put this before the little hemophilia example. So there's your male and female. Shaded means it has the trait. Again, you can always go back and look this up if you have more questions on it. But we can kind of go through it, and I would make sure that you spend a little time looking at it, kind of making sure, does this make sense on how you can kind of figure out who's what, and here you have two carriers and they both passed it on, and so there was a chance of having some of their kids had it, and the sons, it just kind of was random that the sons had it. This one's not sex linked, right? So there's no difference between males and females, but maybe do a little refresher on your pedigrees. For example, is this dominant or recessive? What you should be able to do for tomorrow is you should be able to look at a pedigree and tell me, is it dominant, is it recessive, or is it sex linked?
Recessive.
All right, recessive, because if this was dominant, these two people would be recessive, and you can't have two or all little letters producing a capital letter down there. So that would be recessive. How about this one?
Dominant.
This one would be dominant. Okay, if you want to write that in, that one would be dominant. Think about that. Yeah, you do. Finally, and this is the last slide, your book mentioned, okay, genes are really where things are at, but there's a little bit more to it than just genes. Your phenotype can be controlled by environmental things as well. We all know that, right? Your skin color changes in the summer, perhaps, if you go out. You might get a tan. Some of you get burned. Some of you don't have as much of an effect. If you don't eat very good food, that's going to affect your growth, right? And so phenotypes are affected by both genes and the environment. Again, kind of a minor point on the end.